Now we can discuss that what are the different methods for joining different molecules. But before going to discuss different methods, briefly we can focus that what are different enzymes that can modify the DNA. So different enzymes are there like nucleases, polymerases, reverse transcriptases, and DNA ligases. And in addition to that, terminal transferases, alkaline phosphatases are also there that we are going to discuss later on. Nucleases are the enzymes that play a role in the degradation of the target DNA, and nucleases are also called as DNases. There may be a variety of types of nucleases, but broadly we can say that there are mainly two types, like exonucleases and endonucleases. Exonucleases, they can degrade the DNA at the termini, that either 5' terminus or 3' terminus. So they can start the degradation of the DNA at the ends. In comparison, endonucleases, as their name indicates, they target the DNA from the internal sites. And we just concluded our discussion about the restriction endonucleases, that some can cut the DNA randomly and some at specific points. इसके बाद जो DNA important है वो है polymerases. They may be of different types, like DNA polymerases. They play a role in the replication of the DNA. So DNA polymerases, they can use the DNA as a template and produce another copy of the DNA. DNA polymerases, they may be of variety of type, like type 1, type 2, 3, polymerase 4, polymerase 5. But the most important one, it is the DNA polymerase 3 that is most abundant and the most important in microorganisms. In addition to DNA polymerase, RNA polymerases are also there that can use a DNA molecule as a template to synthesize a copy of RNA by a process which is called as transcription. So, in addition to that, there are also other types as well, which is called as reverse transcriptases. Reverse transcriptases, they are basically RNA-dependent DNA polymerases, and mainly it is produced by RNA viruses. RNA viruses, it is a group of viruses that can infect animal cells, including humans. And they contain the genes for the synthesis of an enzyme like reverse transcriptases. What is its role? तो इसका जो बेसिक रोल है वो ये है कि ये डीएनए को या मैसेंजर आरएनए को एज ए टेम्पलेट यूज़ करता है और इसको कन्वर्ट करता है डीएनए में. आफ्टर कन्वर्टिंग आरएनए इनटू डीएनए, तो जो डबल स्ट्रैंडेड डीएनए है ये होस्ट जीनोम में इनकॉर्पोरेट हो जाता है. आफ्टर इनकॉर्पोरेशन ये इनएक्टिव रहता है या डॉर्मेंट रहता है और ये किसी भी टाइम पे एक्टिवेट होता है. एंड आफ्टर एक्टिवेशन द प्रोसेस ऑफ ट्रांसक्रिप्शन इट इज़ देयर so that messenger RNA is produced, phage coats are produced. So infectious agent, that is viral particles, they are again produced within the cell. So those virus particles that contain this reverse transcriptase, they are called as retroviruses. Another important enzyme, it is DNA ligase. So actually this DNA ligase, it creates a phosphodiester bond between two adjacent nucleotides. So it can catalyze the phosphodiester bond formation between the 5' and that contain the phosphate and 3' end that contain the hydroxyl group. So this DNA ligase, it is very important in the process of gene manipulation.